Not on air is live. Okay. Sorry, I'm not like it's not my actual dog. It's Keith's dog, actually. Oh. I just do it again. It's fine. It's my whole dish. It's just my whole dish. Yeah. Oh my God. Oh, what is going on?
Thank you all for being here. So this is Keith Jacobs' thesis defense, and thank you for joining us, and thank you for supporting Keith during his defense. And I'm going to explain the process in just a minute, but Keith's family is here. So his mother and father and sister are here. Could you raise your hands for me? We'll meet you later after this. But I just wanted to explain to everybody that we're not going to embarrass Keith too much because his family's here and we don't want to embarrass him in front of his family. We're going to have you step out of the room before we start embarrassing. Okay, so a thesis defense is when a student who has worked for 4 years on their doctorate stands before us and tries to defend the work that they have been doing over that past 4 years. For Keith, it's resulted in a remarkably brilliant paper that he has submitted for publication, and I expect will be published very quickly. And so, but before I go on with Keith, I wanted to introduce the key person in Keith's mentoring, and that's Dr. Sharma. So, Gridar, could you stand up for just a second? So, Gridar has Bernard has mentored Keith on literally a daily basis. So even though I'm his thesis advisor, I only get time to meet with him twice a week. But Bernard meets with him many times a day. And his job is to troubleshoot all the technical problems and help them with experimental design and data analysis on a daily basis. And with Bernard, we really thank you and appreciate your mentoring. This process is that Keith is going to present all of his data that supports his thesis that he has presented to his thesis committee. And the thesis committee is sitting right here. Would you have fun raising your hand so that the family knows who you are? So we've got it's typically a committee that consists of about 5 people. And we are professors at Washington University, and we evaluate Keith's progress during the course of the PhD. And they are going to evaluate the presentation today. Then after his presentation, we're going to have a reception out in, so it's on the 3rd floor of the adjacent building. Okay, so let me just tell you a little bit about Keith. Keith has been a real pleasure to work with. He's just a real charming type of person who works well with a team. And he wasn't always that way. When he first joined our laboratory, he had a few problems that we needed to solve. So this is Keith's transition. Keith was born and raised in the backwoods of Long Island. And as you can see from the panel on the left, these are hard data, by the way. There's no arguing these photos. When he first arrived, you can see that he was a little rough around the edges. He drank the wrong beer. He hung out with the wrong people. That young lady in the front. He doesn't hang out with you anymore, right? And he didn't dress her appropriately. But this is the way that people from the backwoods of Long Island behave. So we had our work cut out for us, and you can see that during his transition, he then started hanging out with people that were a little bit more appropriate. Is this young lady in the club? Right. The problem with this transition is that he and she continued to drink the wrong beer. So this is at Blue Ribbon. And so a lot of the people in the audience are from different countries, and I want to teach you some American colloquialisms. There's one of which is called redneck, and another which is called hick. And these are derogatory statements. We don't normally go around calling people that keep redneck or hick. But during his transition, he's really evolved into a brilliant scientist. So what he's going to show you today is some data investigating an extremely important problem in cancer. And so I am, I didn't introduce myself, so I'm Dennis Callahan, I'm the chairman of radiation oncology. And many of the members of the Department of Radiation Oncology are in the audience. Could you raise your hands just so that this family can see how much support Keith has from our department? So a lot of people, right, we're very supportive. The problem that Keith has been studying in the laboratory is a problem that our patients have to deal with for the rest of their lives. We're able to cure them from cancer, but they develop problems that include injury to their brain and to their intestines and bone marrow and many other organs, and people say more about those. And what we want to do is to find out why that happens and then begin to learn how to solve that problem. And Keith has accomplished that, so we are very proud of him, and we will be proud of him forever.
Thank you, everybody, for coming. I'm going to present my thesis work on molecular and epigenetic regulation of normal stem cell radiosensitivity. I performed this work in the labs of Dr. Callahan and Dr. Sharma in the Department of Radiation Oncology. Ionizing radiation, or IR, is a common therapy for treating tumors in cancer patients. IR induces damaging DNA double-strand breaks in order to destroy cancer cells. Unfortunately, cancer cells are rather resistant to radiation, while host tissue can be quite sensitive. This requirement for higher doses results in damaging consequences to normal tissue. Mechanisms of IR-induced normal tissue injury include inflammation, necrosis, fibrosis, and the loss of regenerative capacity. This loss of regenerative capacity produces long-term consequences in several proliferative tissue niches. Undesirable sequelae resulting from damage to these irradiated niches include infertility from damage to the testis, intestinal epithelial erosion from damage to the GI system, hair loss from damage to skin, and cognitive impairment from damage to the brain. The self-renewal of these proliferative tissues is regulated by adult stem cell populations. As many of you are aware, stem cells are capable of undergoing asymmetric cell division, as they can both undergo self-renewal as well as differentiation into a specified target lineage. And these regenerative tissue compartments often comprised of multiple stem cell populations, often both a quiescent and an actively dividing population, which interact with each other in order to regulate the homeostasis of these tissues in response to both injury and normal tissue turnover. The onset of the timing of these consequences that we observe in these tissues parallels the timing of the natural cellular turnover. Therefore, the loss of regenerative potential in these tissues has been associated with the loss of stem cell populations, or as it's commonly known in the clinic, stem cell dropout. Unfortunately, there's been very few studies directly comparing the radiosensitivity of stem cells with their directly differentiated progeny. Most of the studies that do exist only use exclusively non-primary culture models without isogenic differentiation. And of the few in vivo studies that have been formed, these typically only look at a single tissue and therefore cannot get an idea of the overall phenomenon. Therefore, we utilize multiple in vivo stem cell compartments combined with primary culture models in order to identify commonalities underlying differentiation-dependent radiation responses. Here are the in vivo niches that we investigate as part of this project. In brain, we're looking at the subgranular zone stem cell region present within the dentate gyrus, located within the overall hippocampus. In intestine, stem cells are located here in the +4 region in pink, as well as in the base of the crypt interspersed among these Paneth cells. In testis, stem cells are located in the outer spermatogonial region of the seminiferous tubule. And in skin, we are looking at stem cells of the bulb region located about halfway up the hair follicle. We also took much time and effort to establish primary early passage stem cell culture models. Undifferentiated neural stem cells are isolated by dissecting P0 to P2 neonatal mouse hippocampi. And these neural stem cells, or NS cells, can be differentiated by removal of EGF and FGF from media into differentiated neural cells, what we call ND cells. We also established primary embryonic stem cell culture lines, which are isolated originally from the inner cell mass of murine blastocysts. And these ESLs can be differentiated by removal of lip into differentiated embryonic cells, or ED. And we believe that these primary culture systems provide a more realistic model of the true in vivo phenomenon than previous culture models that have been used. So the overall goal of this research is to elucidate the molecular basis for stem cell radiation responses. For this, I have divided this project into 3 aims. For aim 1, I will directly compare the radiosensitivity of stem cells with their directly differentiated isotope project. For aim 2, I'll investigate the contribution of the DNA damage response toward this differential radio response in stem and non-stem cells. And for 3, I'll look at the influence of epigenetic regulation on the DDR. Beginning with aim 1, I will first show data on the relative levels of iron-induced apoptosis on stem and differentiated cells, as well as the effect of proliferation rate on this differential radiosensitivity. Here we have images of irradiated tissue niches from all 4 stem cell compartments. We exposed mice to 6 grays of whole body irradiation. For those unfamiliar with radiation biology, gray is merely a unit of measuring absorbed ionizing radiation dose. And we irradiate these mice using an X-ray machine. Despite the fact that every cell of the body is bathed in radiation, you can see using multiple stem cell markers as well as multiple apoptotic markers, including cleaved PARP, cleaved caspase 3, and tunnel, that at this dose, apoptosis is restricted exclusively to individually labeled stem cells in all 4 tissue niches. Additionally, just about every single stem cell present is undergoing apoptosis, suggesting that indeed stem cell dropout is the likely cause of many of these normal tissue injury effects that we see in proliferative tissues. We were able to recapitulate this phenomenon in both of our culture models. This is a flow cytometry graph for the induction of apoptosis following, once again, 6-gray radiation at various time points. And in both our ES model and NS model, we can see substantial induction of cell death following radiation, while differentiated progeny failed to show any significant increase in apoptosis. We then performed a dose curve on these tissue niches, identifying the levels of apoptosis within the stem and non-stem population. As you can see, stem cells are extremely radiosensitive even at low doses, apoptosing at as low as 2 grays of radiation, while non-stem cells fail to show any induction of cell death until around 8 to 10 grays of radiation. And even at these higher doses, we only see low levels of cell death present. This indicates that stem and non-stem cells have very different thresholds for radiosensitivity, and thus we needed to determine how differentiation state is in fact dictating the relative sensitivity of these cells to DNA damage. A common question that I received early on during the progress of my research was, is it possible that stem cell hypersensitivity to ionizing radiation is merely due to increased proliferation? Well, this is actually based on a long-held tenet of radiobiology, the 1906 law of Burgony and Tribandel, which correlated proliferation rate among various tissues with their radiosensitivity. It's difficult to apply this tenet to stem cell radiosensitivity, however, as stem cells within some tissue niches are quiescent, while others, as I previously mentioned, can be, in fact, rapidly divided. Additionally, it's been suggested that proliferation rate does not actually determine the overall radiosensitivity of a tissue, but merely the kinetics of their radiation responses. Nonetheless, we decided to investigate whether this observed differential radiosensitivity among stem and non-stem cells is in fact due to differences in proliferation rate. So for that, looking at our multiple tissue niches, here I've shown intestine and brain, we utilized Ki-67 staining, which labels actively proliferating cells, co-stained together with stem cell marker. And as you can see, with stem cell marker labeling stem cells in green, only a percentage of actively labeled stem cells are also positive for Ki-67, indicating that some stem cells are actively dividing, while some are not. Additionally, we see a substantial percentage of non-stem cells that are negative for stem cell marker that show positive Ki-67 staining, especially in intestine, as you'll notice, in the upper crypt. These cells are the actively dividing progenitor cells. They're not stem cells. However, their rapid division leads to production of the terminally differentiated cells at the upper part of the villus. This data suggests that increased radiosensitivity in stem cells is not explaining their increased radiosensitivity compared to differentiated cells. This was further confirmed by quantifying the percentage of proliferative cells as compared to apoptotic cells following radiation among SOX2 positive stem cells. As you can see, only a minority of labeled stem cells were indeed actively dividing based on K67 positivity, while the vast majority of these cells were undergoing apoptosis, further indicating that increased radiosensitivity in these stem cells is not associated with greater proliferation. This was also true in our culture models. Here we have BRDU pulse chase, a BRDU pulse chase experiment. BRDU labels actively dividing cells during S phase. When combined with propydium iodide staining for DNA content, we can identify the G1, G2, and S phases. And upon removal of BRDU from the media, we are able to track the cell cycle progression of these cells over time. And as you can see, ES and ED cells both have similar cell cycle progression, completing their cell cycle within around 12 hours. Once again, indicating that stem cells are not proliferating at a faster rate than differentiated cells. So this does not explain differences in iron density. We therefore came up with this model, whereby the law of Burgundy and Trimadel does somewhat explain differences among differentiated cells, as those non-stem cells that apoptosed at 8 to 10 gray that I mentioned earlier were only present within this actively proliferative progenitor region, while terminally differentiated stem cells of the villus were thoroughly radioresistant at all observed doses. However, stem cells, whether dividing or not in the base of the crypt, remain highly radiosensitive at doses substantially lower than any that induce apoptosis in non-stem cells. Therefore, increased proliferation in stem cells cannot be generalized to explain their increased hypersensitivity to ionizing radiation. To summarize aim 1, I have shown you that stem cells are hypersensitive to ionizing radiation, while their differentiated progeny are not. Additionally, this differential radiosensitivity is independent of their proliferation status. Therefore, enhanced radiosensitivity in stem cells must be an inherent property of their undifferentiated state. For aim 2, I am investigating the contribution of the DNA damage response toward this differential radiosensitivity, specifically focusing on DDR signaling, the induction of cell cycle checkpoint arrest and DNA repair, and apoptosis through the MSD1 JANK H2AX pathway. To walk everybody through the DNA damage response, the DNA damage response, or DDR, is essentially a molecular signaling cascade in response to induced double-strand breaks. Upon induction of double-strand breaks, there is recruitment of the MRN complex, comprised of 3 proteins, MRE11, RAD50, and NBS1. The MRN complex senses and detects double-strand breaks, essentially holding the broken DNA in place to allow recruitment of further DDR factors. This is followed by activation and recruitment of PIC proteins, or PI3 kinase-related kinases. These include ATM, ATR, and DNA PK. For double-strand breaks, the most important of these PIC proteins is ATM. ATM can be specifically recruited by MRN to double-strand breaks, where it phosphorylates histone H2X on serine 139 residue to create gamma H2X. Gamma H2X is a major docking site for downstream DDR events. MDC1 can then bind to this phosphorylated serine 139, creating a feed-forward loop together with MRN and ATM to expand gamma H2X for up to a megabase on either side of the double-strand break. This amplifies the overall DDR signaling response, leading to subsequent additional downstream factor recruitment and ultimately activation of DNA repair and cell cycle checkpoints. If the threshold for DNA damage is too high, however, ATM can also activate P53, leading to the induction of apoptosis. So we began in our models looking at the induction of gamma H2X following ionizing radiation. And we observed, first beginning at lower magnification, that in all the stem cell regions, we observed an absence of gamma H2X foci. When we then looked at higher magnification and co-stained with stem cell markers, we saw that indeed stem cells completely failed to show any gamma H2X foci following ionizing radiation. Gamma H2X and the induction of DDR signaling in general is generally assumed to be similarly activated in all cell types. As a matter of fact, gamma H2X is often used as a marker of double-strand breaks itself because it's considered to be so ubiquitous. So this result was extremely striking. We confirm this through quantifying the percent of stem and non-stem cells displaying robust gamma-H2X foci. As you can see, only a small minority of stem cells among these tissue niches display gamma-H2X foci, in stark contrast to their corresponding differentiated cells. We also wanted to confirm whether this phenomenon was true in our culture models. So for this, we utilized subnuclear microradiation. Subnuclear microradiation allows us to induce localized double-strand breaks within a defined region of the nucleus. And we can then observe recruitment of specific DNA repair factors to those localized break sites. This involves incubation of cells for 2 cell cycles in BRDU to sensitize the DNA, followed by irradiation with 405 and 633 nanometer laser. Following subnuclear microradiation, we observed the same phenomenon. In SOX2 labeled stem cells shown here in green, we did not observe any gamma-H2X induced following this subnuclear microradiation, in stark contrast to surrounding differentiated cells, which display robust induction of gamma-H2X. We saw the same phenomenon both in ES-ED model as well as in NSND. Once again, SOX2 labeled stem cells did not display gamma-H2X. We then wanted to look upstream at which step the DNA damage response was first being attenuated. And we observed that both activation and recruitment of phospho-ATM were also attenuated in stem cells. Here we have a Western blot for protein expression. You can see that activation of phospho-ATM based on its phosphorylation at serine 1981 is severely abrogated in stem cells as compared to their ED differentiated progeny. Similarly, phospho-ATM was not recruited by subnuclear microradiation to DNA break sites. Once again, co-plated ES and ED cells. This was also true in vivo, as SOX2 labeled stem cells within the subgranular zone did not display any phospho-ATM foci. So we then needed to observe whether stem cells were even able to sense or detect DNA double-strand breaks in the first place. However, in contrast with this data, we did observe that while gamma-H2X was once again attenuated along double-strand breaks in SOX2 labeled stem cells, MBS1, a member of the MRN complex responsible for detecting these double-strand breaks, was robustly recruited to these break sites, indicating that stem cells are able to initially sense and detect double-strand breaks. And there must be some mechanism blocking downstream transmission of this DDR signal. But before we sought to investigate that specific mechanism blocking downstream transmission, we wanted to determine whether this attenuation of DDR signaling had any functional consequences on cell checkpoint arrest or DNA repair. So we first looked at the induction of G1 checkpoint arrest, which had previously been reported in the literature to be also inhibited in stem cells. We observed a similar phenomenon. By measuring DNA content through rubidium iodide staining, we were able to isolate through flow cytometry G1, S, and G2 populations based on whether cells are before, during, or after DNA replication. We can then track the overall percentage of cells among these various cell cycle phases over time following radiation. And as you can see with the cell cycle profile of ED differentiated cells shown in red, ED cells maintain a substantial G1 population at all time points following radiation, while in contrast, stem cells show a rapid reduction in their G1 population, indicating the absence of G1 checkpoint arrest. This leads to the onset of this substantial G1 population beginning at 12 hours in the ES cells. This indicates the apoptotic DNA fragmentation in these stem cells. This absence of a G1 checkpoint arrest was further evidenced by the fact that ES cells failed to express any P21 protein. P21 protein is a major regulator of the G1 checkpoint arrest, and therefore the absence of any expression would further emphasize the fact that stem cells are unable to induce cell cycle checkpoints due to their attenuation of the DDR. We then moved on to investigating the effect of this attenuated DDR and DNA repair, looking at the recruitment of specific repair factors. We focused on the two major forms of double-strand break repair, non-homologous end joining and homologous recombination. Non-homologous end joining, or NHEJ, begins within minutes and does not require a complementary strand, as it essentially involves ligating together of the two broken ends. Therefore, it occurs primarily during G1 phase and can be error-prone. In contrast, homologous recombination is much slower, taking several hours, as it utilizes a complementary template strand for repair. This means that it can only occur during later S phase or G2M phase. However, it is error-free. When we use phospho-DNA PK as a marker of non-homologous end joining, we observe that it is similarly not recruited to sites of double-strand breaks, similar to gamma-H2X following subnuclear microarration of stem cells, in contrast with surrounding differentiated cells. Phospho-DNA PK foci are also not seen in vivo within SOX2 labeled stem cells with dentate gyrus. For homologous recombination, we utilized RAD51 as a marker of this repair pathway. And both in individually labeled stem cells of testis and the entire stem cell region of the intestinal crypt, we do not see the presence of any RAD51 foci, once again in contrast with surrounding differentiated cells. You'll also note that in intestine we see robust RAD51 foci present once again in this progenitor region, which I previously showed was actively proliferating, however, was thoroughly radioresistant. To investigate the actual effect on DNA repair efficacy, we utilize the neutral comet assay. The neutral comet assay is single-cell gel electrophoresis following induction of DNA damage and nuclear lysis. In this example of an unirradiated cell, you can see that because the DNA is intact following electrophoresis, the majority of the DNA remains in the head of the comet. However, following DNA damage, where there are several smaller DNA fragments present, we see migration creating somewhat of a comet tail. We can quantify the length and density of this comet tail using specific software, give us a factor known as olive moments. By measuring the reduction in olive moment over time, we can observe the progression of DNA repair. And as you can see here, differentiated cells show rapid and efficient induction of DNA repair, while in contrast, stem cells show only a mild DNA repair kinetics, and they fail to complete DNA repair by 4 hours, the time point at which they begin to drop out and apoptose. So having shown that stem cells exhibit attenuated DNA damage response, and this results in attenuated cell cycle checkpoint activation and muted DNA repair, we would expect these stem cells to apoptose. However, as I showed you earlier, apoptosis typically involves signaling through the DNA damage response, specifically through ATM and p53. However, p53 has been shown in the literature to not be responsible for iron-induced apoptosis in stem cells. And I've shown that ATM is not activated in these stem cells. To confirm this, we irradiated cells in wild-type mice and compared them to dentate gyri of mice from H2AX and ATM knockout mice. And as you can see, based on cleaved PARP staining, there is very comparable levels of apoptosis in the stem cell regions of all 3 mice, indicating that iron-induced apoptosis in stem cells is likely independent of DDR signaling. Therefore, there must be some alternative apoptotic pathway present in stem cells that does not require the DNA damage response. Some evidence leading us in the correct direction to identify this pathway was the fact that in this image I showed you earlier, whereby at 15 minutes following the radiation, stem cells failed to display any gamma-H2X foci, which I'll remind you is phosphorylation at serine 139 histone H2X. Using the same antibody, when we look later at apoptotic time points, we now see a singular pan-nuclear phosphorylation in SOX2 labeled stem cells, suggesting that there is somewhat of a biphasic regulation of this serine 139 phosphorylation, whereby they are unable to activate gamma-H2X foci as part of the DNA damage response. However, during apoptosis, we observe this pan-nuclear serine 139 phosphorylation. So we then search for potential apoptotic pathways involving this pan-nuclear serine 139 phosphorylation, and we came upon the MST1 JANK H2X pathway. MST1 is mammalian C20Y kinase 1. a member of the Hippo pathway that is involved in controlling organ size during development through its role in apoptosis. MST1 is cleaved by caspase-3 following apoptotic stimuli. In addition to caspase cleavage, it is phosphorylated, leading to its nuclear translocation, resulting in its full activation. Fully activated MST1 can phosphorylate several targets, including histone H2B at serine 14, as well as JANK. JANK is C-gin N-terminal kinase, and together with MST1, it can phosphorylate histone H2X at this same serine 139 site that is targeted by ATM. This pan-nuclear phosphorylation of H2X is required for the recruitment of caspase-activated DNases, which are involved in apoptotic DNA fragmentation and condensation, the end stages of apoptosis. So we then looked for involvement of MST1 and JANK in this pathway, and we observed that activation of MST1 and JANK based on their specific phosphorylation sites is indeed active only in stem cells at later apoptotic time points, while differentiated cells do not show any phosphorylation of these apoptotic proteins. We then performed a time course over time on these in vivo compartments following 6-stray radiation. And at early time points, we observed similar to what we saw before, where serine 139 phosphorylation is present in the form of gamma-H2X foci, while MST1 is only cytoplasmic. However, at apoptotic time points, we now see once again the singular pan-nuclear phosphorylation corresponding to nuclear translocation of MST1, as is expected based on the pathway. We additionally observed that phosphorylation and recruitment of JANK is only present within individually labeled stem cells of the dentate gyrus, and this phosphorylated JANK co-stains with pan-nuclear serine 139, once again in stem cell leaf. Together, this data indicates that iron-induced apoptosis in stem cells does involve the MST1 JANK H2X pathway and is likely DDR independent. To summarize this section, I've shown that stem cells exhibit an attenuated DNA damage response that results in muted cell cycle arrest and diminished DNA repair efficacy. This iron-induced apoptosis in stem cells involves the MST1 JANK H2X pathway and does not appear to involve the DNA damage response. So based on this data, it appeared that stem cells are forgoing even the initiation of DNA repair signaling and being almost shunted immediately towards apoptosis. So we are curious whether there was some pathway or mechanism we could identify that is directing these cells towards apoptosis in lieu of even attempting DNA repair. To that end, this brings us to aim 3, the influence of epigenetic regulation on the DNA damage response. And we focus on phosphorylation of tyrosine 142 on histone H2X and acetylation of H3K56. There was some great work done by the Rosenfeld lab in 2009, which identified a DNA damage responsive residue on histone H2X adjacent to this serine 139 site. This tyrosine 142 or Y142 is regulated inversely with S139, as Y142 is phosphorylated under baseline conditions, and following DNA damage is dephosphorylated, paralleling phosphorylation of H2X at serine 139. This dephosphorylation is required for NDC1 binding and amplification of the DNA damage response, as when the phosphatase EYA was removed from these cells, dephosphorylation was inhibited, MDC1 binding was blocked, and instead of DNA repair, instead these cells apoptose through recruitment of JANK. Since I have previously shown that stem cells apoptose through JANK recruitment and do not signal through the DNA damage response, I was curious whether unique regulation of H2X tyrosine 142 phosphorylation could be dictating their responses to DNA damage. So we utilized this subnuclear microradiation, and beginning in our differentiated cells, we see a response as predicted by the model, where there's almost a complete mutually exclusive relationship between tyrosine 142 phosphorylation and S139 phosphorylation, or gamma-H2X. In contrast, stem cells fail to dephosphorylate tyrosine 142 in following subnuclear laser microradiation, corresponding to their attenuation of gamma-H2X induction. And here, even on a global level, we can observe that tyrosine 142 phosphorylation is only inhibited in differentiated cells. And just on the right here, this is a image of co-plated ESED cells where we see the same phenomenon, dephosphorylated non-stem cells, but not in stem cells. As expected, also based on this model, MDC1 is similarly not recruited to laser tracks in stem cells in contrast with surrounding differentiated cells. And in stem cells, this MDC1 recruitment corresponds to complete dephosphorylation of this tyrosine 142. We therefore hypothesize that persistence of H2AX phosphotyrosine 142 may be blocking MDC1 mediated expansion of the DNA damage response and could be the mechanism directing stem cells toward apoptosis. Having established that unique histone modifications and epigenetic regulation in stem cells appears to be influencing their DNA damage response, we chose to investigate the influence of the overall stem cell epigenetic profile. Several studies have identified both beneficial and detrimental effects of various histone modifications on DDR signaling in both yeast and mammalian cells. Specifically, histone 3 lysine 56 acetylation, or H3K56 acetylation, has been identified as an important regulator of the DNA damage response. Additionally, H3K56 acetylation is enhanced in undifferentiated stem cells. On the left here, we have a graph showing that in the human ES lines, HSF1 and HSF6, K56 acetylation is substantially enhanced compared to two somatic cell lines. And on the right here, this Western blot shows that K56 acetylation rapidly decreases upon differentiation in Drosophila cells. So when we looked in our model, we observed similar results. H3K56 acetylation was greatly elevated in the inner cell mass of murine blastocysts, where ES cells are derived from. We also observed that H3K56 acetylation corresponds to SOX2 staining and is quickly reduced upon differentiation of these ES cells. We then co-plated, once again, ES and ED cells in culture and irradiated them. However, this time we co-stained for H3K56 acetylation and gamma H2X. We observed that these cells which failed to display gamma H2X foci, indicating an attenuated DNA damage response, were the cells that displayed elevated H3K56 acetylation, suggesting that increased acetylation in stem cells may be inhibitory for their DDR signaling. We also confirmed that this was true once again in vivo, as in these stem cell regions where gamma H2X foci are not present, we observed elevation of H3K56 acetylation. We then wanted to determine what the response of K56 acetylation is along break sites, similar to the experiment I just showed you using subnuclear microradiation with the phosphotyrosine 142. This experiment has actually been performed by the Steve Jackson lab back in 2010, albeit on cancer cells. And on these two cancer cell lines, you can see that H3K56 is completely dephosphorylated along gamma H2X labeled tracks of double-strand breaks, suggesting that K56 acetylation may be inhibitory for DDR signaling, and its downregulation is required to allow induction of gamma H2X and the DDR pathway. So we performed subnuclear microradiation once more on our ES and ED cell models. And we observed that in non-stem cells, as similar to what was shown in cancer cells, we see a stark reduction of H3K56 acetylation along gamma H2X labeled tracts of DNA damage. While in stem cells, which already display thoroughly elevated H3K56 acetylation levels, we do not see any reduction along break sites. This was quantified by measuring the signal intensity along the double-strand break laser microradiation tract. You can see in ED cells that in the tract of DNA damage where gamma H2X is rapidly induced, we have a corresponding drop in K56 acetylation. While in stem cells, we only have mild induction of gamma H2X and no real response whatsoever in H3K56, certainly not any reduction at the site of double-strand breaks. Interestingly, when we looked at later time points, at 30 minutes and 1 hour, as we previously only observed this reduction in ED cells at 10 minutes. At these later time points, we've now failed to see any reduction in acetylation levels in these differentiated cells, suggesting that there's a very transient relationship between K56 acetylation levels and the DDR. This agrees with what's been suggested in the literature that K56 acetylation must be reduced to allow the initiation of DDR signaling. However, its restoration is required to allow the cell to restore the basal chromatin state following the completion of DNA repair. We then wanted to determine whether we could make a more causal connection between this K56 acetylation and the inhibited DDR in stem cells. For that, we needed to be able to modulate K56 acetylation levels. To do that, we needed to target its regulators. The options were among the histone acetyltransferases and the deacetylases. The histone acetyltransferase is most primarily associated with H2K56 or P300 and the closely related CBP. Histone deacetylases associated with H2K56 are SIRT-T12 and HDAC12 among others. We chose to focus on targeting P300 due to its increased specificity towards the H2K56 residues as opposed to other histone residues. Additionally, P300 has been shown to promote iron-induced apoptosis, as cell death was inhibited in P300 but not CBP-deficient MCF7 cells. P300 also activates the master apoptosis regulator P53, and it's been shown to be lost in several cancers associated with the promotion of tumor progression. Therefore, inhibition or knockdown of P300 may improve the radiosensitivity of stem cells, presumably through its effect on H3K56 acetylation. We observed through both immunofluorescence and protein expression on Western blot that P300, similar to H3K56 acetylation, is greatly elevated in SOX2-labeled stem cells and decreases upon differentiation. And this increased P300 expression remains following irradiation. Therefore, P300 acetyltransferase may be responsible for the observed elevated H3K56 in stem cells. We were then able to determine that siRNA knockdown of P300 could downregulate levels of H3K56 acetylation. You'll also notice that this was also very temporarily regulated, as by 48 hours, we now see restoration of the H3K56 acetylation mark, likely through to compensation from other acetyltransferases. So in order to get more precise temporal control of this downregulation, we also combine this with use of the ICBP-112 bromodomain inhibitor. Bromodomain is a region on P300 which mediates its interaction with the H3K56 target residue. And by inhibiting this interaction, we block phosphorylation of target sites. We can see that following only 30 minutes of incubation of this drug, we once again get robust downregulation of this acetylation mark. Through these two assays, we were able to determine whether downregulation of H3K56 acetylation could have an impact on DDR signaling in stem cells. We did successfully observe that following P300 knockdown as compared to negative siRNA control, we see strong increase in gamma H2X induction following radiation. Similarly, following incubation with this P300 inhibitor, we see that K56 acetylation is decreased, corresponding to increased density of gamma H2X foci in the ES cells. More importantly, knockdown of P300 and downregulation of H2K56 acetylation also resulted in improved DNA repair. This is once again a COMET assay. You can see whereby reduction in olive moment represents progressive DNA repair. You can see that following P300 knockdown, we have improved DNA repair as compared to negative siRNA control. This determines a causal relationship between H2K56 acetylation and DDR signaling, indicating that reduction of acetylation in stem cells can restore DDR signaling and improve DDR repair. More importantly, P300 knockdown reduces iron-induced apoptosis in stem cells without any effect on differentiated cells, determining that H356 downregulation can specifically protect stem cells from iron-induced apoptosis. To summarize this final aim, I have shown that persistent H2AX tyrosine 142 phosphorylation around break sites in stem cells may direct them to apoptosis in lieu of repair, and constitutively enhanced H356 acetylation in stem cells directly influences their radiosensitivity through the DNA damage response. Thus, cellular responses to DNA damage appear to be heavily influenced by their epigenetic profile. Just to summarize all the data that I've shown today, we observed that stem cells are able to detect double-strand breaks through recruitment of the MRM complex, specifically NBS1. However, downstream DDR signaling is attenuated through phospho-ATM, gamma-H2X, MDC1, as well as DNA repair factors, DNA PK, and RADP1. In contrast with non-stem cells, which show strong DDR signaling through all of these factors. This results in the induction of apoptosis through various factors, including cleave-PARP, cleave-caspase 3, MSD1, and phospho-J, resulting in pannuclear phosphorylation of histone H2X, the same site targeted through ATM as gamma H2X. These radio responses are associated with unique epigenetic regulation, specifically persistence of tyrosine 142 phosphorylation along double-strand breaks, and constitutive elevation of H256 acetylation. Overall, from this data, we can conclude, as I mentioned previously, DDR is not nearly as ubiquitous as has been currently assumed. And we cannot generalize that responses to DNA damage will occur similarly in all cell types. And we've shown this through the fact that unique epigenetic profiles can clearly lead to vastly different responses to DNA damage. Even more importantly, modulation of specific histone modifications may provide therapeutic potential for minimizing iron-induced normal tissue injury. And this work on H356 acetylation works as a proof of principle that we are capable of specifically targeting stem cells for reducing their radiosensitivity without any effect on other cells. For future directions, I plan to more thoroughly investigate the effect of H356 acetylation modulation on DDR signal. Specifically, I'll look at any potential contributions of CBP, the related acetyltransferase 2P300, as well as ASF1, the histone chaperone that recruits P300 and CBP to target residues. We'll assess differences in DDR factor recruitment and activation following downregulation of H356 acetylation. I would also be interested to determine whether upregulation of this acetylation marking differentiated cells can actually increase their radiosensitivity and reduce their DDR signaling. I'd like to thank all the members of my lab, especially Dr. Allen and Dr. Sharma. Also Barbara Meyer, who did a lot of the work I've shown today. Very happy she decided to join the lab because I could not have completed all this without her. Also, thanks to Sherry, Suyach, and Katie, along with Sandeep, former member of the lab, and everybody else from the Hallahan lab who's helped me over the years. Also need to thank Bob and Jesse from the Department of Radiation Oncology who helped me with various technical aspects of this work, as well as my friends Chris and Hugh who helped with ES and NS culturing respectively, and Ann Benz and her PI, Dr. Steve Menerick, who taught us how to dissect the hippocampus of mice. Most importantly, thank you to my thesis committee for all your advice and help over the years with this project, Barry, Susanna, Kristen, and Leela. Thanks to all of you. And thanks to funding through the NIH Site and Cancer Center and the Department of Radiation Oncology. I'd just like to take an extra moment to talk about my two PIs. Dr. Hallahan has been amazing over the years. I'm always impressed by the fact that despite being department chair of a clinical department where he oversees residents, physicians, all the members of his lab, and several students, he's always available. He's able to see us several times per week. He's helped me thoroughly with all the important steps of my PhD, and I thank him immensely. Godar, I basically see every day. He's the brainchild of this project, basically. And while we haven't always been on the same communication wavelength at times. I know that I thoroughly appreciate his passion, all the effort he's put into ensuring that I have a successful PhD and that I'm doing good science. And I owe the majority of what I've learned during my grad school time to him. I also need to want to just take some time to point out some extracurricular academic organizations that helped me. Starting in high school with Science Olympiad, learning the value of troubleshooting and trial and error did great wonders to help me get through grad school. Additionally, I had great experiences with the Young Scientist Program and the Student Consulting Group BALSA. Also, for 3 years, I was involved in the medical school musical. This was a great way to blow off steam and be a little silly. First beginning as an actor, this is an old racist man, which I guess goes with Dr. Helen's introduction to me. And then last year, moving on to being producer, I had tons of fun being on the production team, and everyone was awesome. Also, the rest of my friends in BBBS, there's a picture from our classy Christmas party. Grad school was some of the best years, definitely, I've had in my life. And it was great having people to similarly nerd out with, party with, complain at times in the rare times when necessary, whatever we needed. And thanks to all of you for your help over the years. And I definitely could not have had such a positive grad school experience without all of you. So thank you. And also, this is Bob Mercer putting me in a headlock. I especially want to, despite her strong protest, I also have to point out, thanks to my girlfriend Gina, she was unfortunate enough to start dating me during the most stressful time of my grad school career, but her patience and ability to keep me relaxed has done wonders to help me get through this, so thank you for all that. I also need to especially point out to my closest friends, Tiffany, who I was friends with before I even was accepted to WashU, met during interview weekend, Kara, who you already saw earlier, Beth, Chris, and especially Kevin, my partner in argumentative related crime. It's a picture of us in a more joyous moment. And most important of all, my family. I'm very happy that my sister was able to make it here today. It's her first time in St. Louis, so I'm glad she was able to see my presentation. And I had to include this picture to embarrass her. I think 5 years old. This is adorable. Of course, most important, thanks to my parents. Everything I am today is due to them. Of course, not just intellectually, but just as a person. And thanks for supporting me along the way with whatever I needed. And to my family overall as a whole. And thanks to everyone else who was here. And I'll take any questions after I conclude with this.
Point is ask questions from the audience before we have you exit, and then the committee will ask these few questions related to so let's just open it up and find out if there are any questions from graduates.
Kevin.
Yeah, thinking broadly, it's not my field, but I was wondering what might be, obviously, stem cells is live after we are going to go straight to speak on what might be advantageous broadly for that process, narrowly or
It's most often cited in the literature. Of course, this is just theoretical when we're talking about evolutionary reasons. Most often discussed that it could be beneficial for stem cells if they're exhibiting any type of damage to apoptosis, so you're not passing on damage to further progeny of the tissue. However, in nature, you're never really exposed to 6 rays of radiation, so there's not much evolutionary pressure to be able to really respond to this level of DNA damage. So we think that while there might be some benefits to a stress response in terms of stem cell dropout, we think overall really it's a coincidence of this epigenetic profile that defines their pluripotency. Just as it restricts transcription factor recruitment, it also happens to restrict DDR or DNA repair factor. Daniel?
So the H3K56 and H300 differences that's pretty striking between stem cells and the different function. Can you comment on what else that epigenetic modification is doing?
So part of the reason why it's so striking is because this acetylation mark is especially elevated along the promoters of pluripotency genes. It's overall, in terms of transcription at least, an activating mark for transcription. So it basically promotes transcription of pluripotency genes such as SOX2 and DOX4. So that's primarily why we have such constructive. Both P300 and K56 have been shown to be substantially involved in maintaining the stemness of the stem. Yes.
So your reversals are mostly from the differentiated cells. Do you look at all at how the transition?
No, we would love to look on. I actually have done some preliminary data on P300 knockdown in cancer cells, medulloblastoma cells. And we do not see any effect on radiosensitivity. We think it's because there's basically a threshold level for acetylation, and if you're under that threshold, you can readily respond fine. But we have not been able to look at cancer stem cells, which would be the most interesting, as they exhibit kind of a radio-resistant phenotype, but an undifferentiated phenotype. And that we hope to look at in the future.
Thank you. We don't want you to